The principal sites of immunoglobulin A synthesis and secretion are the gut, the respiratory epithelium, the lactating breast, and various other exocrine glands such as the salivary and tear glands. It is believed that the primary functional role of immunoglobulin A antibodies is to protect epithelial surfaces from infectious agents, just as immunoglobulin G antibodies protect the extracellular spaces of the internal tissues. Immunoglobulin A antibodies prevent the attachment of bacteria or toxins to epithelial cells and the absorption of foreign substances, and provide the first line of defense against a wide variety of pathogens. Newborn infants are especially vulnerable to infection, having had no prior exposure to the microbes in the environment they enter at birth. Immunoglobulin A antibodies are secreted in breast milk and are thereby transferred to the gut of the newborn infant, where they provide protection from newly encountered bacteria until the infant can synthesize its own protective antibody. Immunoglobulin A is not the only protective antibody a mother passes on to her baby. Maternal immunoglobulin G is transported across the placenta directly into the bloodstream of the fetus during intrauterine life. Human babies at birth have as high a level of plasma immunoglobulin G as their mothers, and with the same range of antigen specificities. The selective transport of immunoglobulin G from mother to fetus is due to an immunoglobulin G transport protein in the placenta, FcRn, which is closely related in structure to MHC class I molecules. Despite this similarity, FcRn binds immunoglobulin G quite differently from the binding of peptide to MHC class I, as its peptide binding groove is occluded. It binds to the Fc portion of immunoglobulin G molecules. Two molecules of FcRn bind one molecule of immunoglobulin G, bearing it across the placenta. In some rodents, FcRn also delivers immunoglobulin G to the circulation of the neonate from the gut lumen. Maternal immunoglobulin G is ingested by the newborn animal in its mother's milk and colostrum, the protein-rich fluid secreted by the early postnatal mammary gland. In this case, FcRn transports the immunoglobulin G from the lumen of the neonate gut into the blood and tissues. Interestingly, FcRn is also found in adults in the gut and liver and on endothelial cells. Its function in adults is to regulate the levels of immunoglobulin G in serum and other body fluids, which it does by binding circulating antibody, endocytosing it, and then recycling to the cell surface. Have a great day.